screen?
Super.
Okay, great.
Great. Well, thank you very much, Anne-Flo, and thank you to all the organizers for the opportunity to speak, and also for your patience while I vacillated this week in debating whether to give a new talk or go with something a little bit more well-established. I decided to give a new talk. So we'll see how that goes. I also want to thank everyone in the audience for sticking around to the end of the day, and for your forbearance on a new talk. Before we get into the science, let me acknowledge the people that have made this possible for me. The first is Dave Morgan, my grad student, who's running all the experimental work that I'll describe for you. And the other is Lauren Crawford, a really gifted biostatistician at Brown, now also at Microsoft Research, who helped me a lot in the early development of some of the ideas that I'll be presenting today. And finally, this wouldn't be happening without support from the National Science Foundation. So here's my plan of attack. What is epistasis and why it matters scarcely needs to be reviewed, but I have a few comments here just to sort of maybe orient you to my personal obsessions around epistasis. Then I want to introduce a technology that also doesn't need a lot of introduction, deep mutational scanning, which has been used very effectively to learn about fitness effects of individual mutations. And then we'll get to the new work, an idea that we've had to use pairs of deep mutational scanning experiments to learn something about epistasis. We don't have any data of our own yet, but we were fortunate to find a data set in the literature from Jesse Bloom's lab that allowed us to sort of pressure test some of our analytic ideas. But at the end of the talk, I'll describe the work we're doing in the lab and also highlight what I think of as the challenging open analytic theoretical questions. Okay, what is epistasis and why does it matter? Epistasis, of course, means that the consequence of a mutation depends on its genetic background. A little bit of history. I'm not quite as old as William Bateson. This term was coined at the turn of the previous century to describe deviations from Mendelian expectations in dihybrid crosses. So this is just a quick schematic to see how that works. This actually then teaches us, has taught us a lot about the way biochemical pathways work. Imagine I have a pigment, a flower that's blue. This actually is a flower that's blue, and it gets its blue pigmentation through this two-step enzymatic reaction that converts a white substance into a magenta substance and then into the blue. And so if we imagine a dihybrid cross, this is the sort of thing Bateson was thinking about, and we count how many, what proportion of the offspring have each of the phenotypes, what we observe is that this cell here, which is missing both genes, is actually looks the same as the cells of the organisms around it, because losing the second enzyme is immaterial in the genetic background that's already lost the first enzyme in this pathway. From my own perspective, the importance and excitement around epistasis has to do with its influence on adaptation. The language that I use has to do with fitness landscapes, and I would say we can agree epistasis induces non-linearities on the fitness landscape. I'll say a little bit more about that in a minute. And consequently can perturb the path that natural selection, the paths that natural selection can take. So this is probably the figure that I'm most well known for. This is a paper I wrote while I was still a postdoc with Dan Hartle. In this study, we constructed all combinations of an antibiotic resistance gene in bacteria, all combinations of 5 mutations that were known to collectively increase drug resistance 100,000-fold. And we characterized the drug resistance of each of these combinations, and were able to show that there are many cases where the epistasis among these mutations is so extreme that mutations that are beneficial in some genetic backgrounds in the context of some mutations are deleterious in others, so that natural selection has to be, is constrained in the temporal order in which mutations will be favored. This is a picture from a recent review of Martin's. He actually mentioned this paper an hour ago, just to point out that epistasis will induce these correlations in sequence alignments. This is important if you're a phylogenetics-minded researcher, because of course, many methods originally assumed independence among sites. Martin's group and the work he talked about today takes that problem and turns it into a tremendous virtue by making inferences about these patterns of interaction to be able to understand patterns of interaction from patterns of correlation. For me personally, the biggest takeaway from the paper in TEM1 beta-lactamase was an appreciation of what might be described as the combinatorics of epistasis. Let me say what I mean by that. In principle, any subset of loci in the genome can interact epistatically, and the number of subsets of loci in the genome grows exponentially in the number of loci. Or in the context of the experiment I described, and others have done similar experiments, if I'm interested, as I was, in 5 mutations, the number of constructs I have to build is 2 to the 5th, and that, or 32, which then defines that number of interactions. There will be, in my case, 5 first-order effects, the effects of each mutation in isolation. There'll be 5, choose 2 or 10 pairwise effects, because I can construct 10 pairs of mutations, and so on. This shouldn't surprise us. It's not that this accounting of epistatic terms reveals a complexity that wasn't present formerly. I just told you that I had to construct 32 combinations in order to get the data to begin with. And what epistasis does is really just convert those observations into a like number, those phenotypic observations into a like number of epistatic observations. Now, do those epistatic interactions matter? And certainly, going back to Martin's talk and other talks in this program, and there's good evidence for the importance of pairwise interactions, and Martin made that point very clearly. This is a study from Roy Kishoni's lab of a few years ago that really struck me. What Roy and his group did was an experiment that was analogous to the one I did. They were interested, in fact, also in drug resistance evolution, also in E. coli, a different enzyme and a different drug. But they constructed all combinations of, in this case, 6 mutations, characterized the drug resistance, and then converted those numbers into epistatic terms. And then they said, let's take all those epistatic terms and rank them in order of magnitude, in order of importance of explanatory power. And let's build a model that attempts to predict the drug resistance of each of the combinations of mutations in their data, using only the largest effect epistatic term, or the largest and the second largest, or the first and the second and the third largest term, and so on. So that's what this figure is showing us. Left to right is a model of increasing complexity by the inclusion of ever more epistatic terms. And importantly, the first term included was the one of largest effect. And the y-axis is the residual variance. It's the amount of the variation in drug resistance phenotype that couldn't be explained by the model. And what you see is it drops quickly. It's a log scale on the y-axis. But the thing that really jumped out at me and my collaborator, Robert Heckendorn, is how many higher order interaction terms are present at the left end of this figure. So the first the mutation of biggest effect is a single mutation in the promoter region. That's the 35C to T. The next most important explanatory mutation, epistatic term, is a 3-way interaction. Then comes a 2-way interaction, and then come a series of 3-way interactions. And what Robert and I wondered is, should we be surprised? Because after all, the combinatorics that I outlined, as represented here, tell us that there are more high-order interactions than there are pairwise or first-order effects. So the question that Robert and I asked is there an overrepresentation of high-order epistatic terms in this data set? That is to say, are large epistatic terms enriched for higher order epistatic terms, above what you'd expect by a random permutation where you just randomly assign interaction terms with respect to their frequency, given in the histogram on the right, from left to right in a figure like you see on the left. In order to answer this question, Robert and I took advantage of the fact that at that time there were 15 or 20 other data sets that were like this one and the one that I prepared years ago, and what we call combinatorially complete, which means that the experimentalist went to the trouble of constructing all combinations of some set of mutations. And so we could estimate the epistatic coefficients of all order from these data sets. And indeed, across data sets, there's a very strong signal that higher order epistatic terms are overrepresented for their explanatory power. Okay, that's by way of introduction. I remain interested in understanding empirical patterns of epistasis. And so now I want to briefly review a technology called deep mutational scanning that's been used to develop unbiased estimates of the distribution of fitness effects of single mutations, and then talk about how we plan to use those same techniques to estimate pairwise and higher order interactions. So what is deep mutational scanning? The idea here is I have an enzyme or some protein that affects the ability of a microbe to grow and reproduce. And using trickery of molecular biology, I can construct a library that's every single point mutation off of some reference sequence, some wild type sequence. So for example, the data set of Fernberg's in TEM1 beta lactamase was already mentioned. That was something that Martin built a piece of his talk around. And what those authors did was to build every single one-off mutant from TEM1 beta lactamase, put the entire library into E. coli cells, expose the cells to ampicillin, and watch the change in frequency. So schematically, maybe there are 3 variants in this schematic picture: blue, green, and purple. After selection, the purples have gone up in frequency, the greens have gone down, greens have unchanged, and the blues have gone down. And the change in frequency is some kind of proxy for fitness. So this is one of the first figures in Fernberg's paper. This is the entire TEM1 beta lactamase enzyme. Well, it's the first 70 residues arranged left to right. So each column is a position in the enzyme, and each row is a different amino acid. And the heat map tells you the fitness effect with respect to drug resistance of that particular mutation in isolation. And what they produced was this figure, which I find really satisfying, and because it's an estimate of the distribution of fitness effects that's unbiased by natural selection. When we go to nature and look at what sequences we see and what frequency, we're watching the convolution of the distribution of fitness effects modulated by selection. And this technology is before selection acts. So what we plan to do is what we're calling paired deep mutational scanning. This is a schematic of what any single deep mutational scanning exercise looks like. The x-axis is the number of mutations, the Hamming distance from the starting allele. The y-axis is some kind of schematic representation of the number of variants at each Hamming distance. It's log transformed to reflect the exponential growth and the number of mutants as you move away. The entirety of the deep mutational scanning library, however, is located at Hamming distance 1. So because we're only observing each mutation on one background, we don't know anything about the epistasis. The idea wasn't very complicated. I thought, let's do a second deep mutational scanning exercise on a homolog of the first. Now we start to see mutations on on 2 different backgrounds. And so there must be a lot of information in that kind of paired data set that speaks to the patterns of epistasis in this system. The first and most obvious way of going after these data, or the first and most obvious thing to do with these data, is to compare the effects of all missense mutations at each residue that's identical in the 2 backgrounds. So imagine, for example, focusing on the first codon, the methionine, and I can mutate it to any of the other 19 amino acids, or indeed even to a stop codon. And then I can make a scatter plot where the y-axis is the effect of that mutation on the background 1, and the x-axis value is the effect on background 2. So here are some asterisks that I scattered onto this, these axes. Any mutations that lie on or near the 1-to-1 line exhibit no epistasis because, be they beneficial or deleterious, their effect on the two backgrounds is the same. And what we'll be interested in is how many of these points are significantly off of the 1-to-1 line. This is one approach for trying to learn something about epistatic signals in these paired deep mutational scanning data sets. As I said, we don't have any data of our own yet, but fortunately, a couple of years ago, Jesse Bloom's group published a lovely study in which they did a deep mutational scanning exercise on 2 alleles of the envelope protein in HIV. They were able to look at 616 amino acid residues, 92 of which differ. So they have an exhaustive sampling of radius 1 around each of these 2 backgrounds called BG505 and BF520. And again, we're going to run through the 524 positions that are identical in those 2 alleles, multiplied by 19 missense mutations, and make scatter plots of the sort I've already shown you. So my student David Morgan did exactly that, and the Haddock's paper reports estimates of experimental error for the fitness effects. And so David used that to estimate, to be able to assess whether any particular point in any particular of those scatter plots, remember there's 1 per position in the protein, 1 per position where the 2 alleles are identical. Ask whether those points are significantly off the 1-to-1 line. And after correcting for Bonferroni, you can see here the x-axis is the protein, the y-axis is the opposite log 10 p-value. So in this Manhattan plot, the red line is the relevant Bonferroni line. There's 1 mutation that's significantly off the 1-to-1 line, which is a little bit disheartening, or put another way, tells us something about the kind of experimental error that we need to strive for in our own experiments. Nevertheless, there turns out there's a huge overrepresentation of very small p-values shown here on this QQ plot. So if there were no epistatic signal at all in these data, we would expect a uniform distribution of p-values, and there's a huge, huge excess of very small values. So that's somewhat heartening that this approach produces some kind of signal, even only indirectly. Okay. So this brings us to the current moment, current work, and open questions. And I'm particularly excited to be presenting to this community because there are some important open questions that perhaps some of you will have some thoughts and suggestions for. So first to the lab, and this is what David does in his spare time. I've already alluded to the Fernberg data set, a deep mutational scanning library in TEM1 beta-lactamase. Mike Stifler, whom Martin also mentioned, produced a similar data set. Olivier's lab also produced one earlier in the last decade. So what we've decided to do is, rather than reinventing the wheel, we're building a deep mutational scanning library in SHV1, which is the closest natural relative of TEM1. So here's a phylogeny of this class of beta-lactamases, and you can see here that TEM1 and SHV1 are each other's closest relatives. For me, the work is not experimental, it's theoretical, and the kinds of questions that I've been wrestling with fall into 2 or really 3 categories. So the first one is, what Hamming distance between the 2 backgrounds will be most informative? So this is a schematic, again, of essentially one of the points in the scatter plot that corresponds to the first residue in the data we anticipate. This is showing, this is asking the question, what's the effect of converting methionine to cysteine, the methionine at position 1 to cysteine on the TEM1 background and on the SHV background. And these two starting points differ at 91 amino acids. That's given to us. We don't have a choice in turning that knob, but it does seem like there's some interesting work to be done in thinking about where the sweet spot is for getting the most information out of these kinds of data sets. And this is, I think, very closely related to the point that Martin made late in his talk, that if, in a somewhat different context, that if the 2 backgrounds are too similar to each other, or in the language of that piece of his talk, if the evolution experiment isn't run for long enough, then there isn't the opportunity for epistatic signals to build up. At the other limit, I think we come to the place that one of the questions was motivated by, which is once the two sequences become too far diverged, everything is epistatic because the proteins really stop being in any meaningful sense orthologs. And so we shouldn't be surprised that every mutation does something different in the two backgrounds. I don't really know how to tackle this question. I have some ideas for reasonable models of fitness landscapes and how to sort of explore this computationally, but I haven't made any progress on it. The other question is how to get the most out of the data once we decide how far the two background alleles ought to be from each other. To get to that, for me, the question is really comes to where do the epistatic effects live in these data? So I showed you one approach, which is to ask what's the effect of the same mutation on the two backgrounds. I want to be a little bit more comprehensive and principled in thinking about where else to look for data. And to get there, I need to do a little bit of development using the nomenclature of the sequence space. So again, a concept that probably doesn't need much introduction to this audience. Sequence space represents, is a way of representing all combinations of mutations in such a way that mutationally adjacent combinations are spatially adjacent. So we assign one locus, we assign one dimension in this space to each locus. This is a one-dimensional sequence space, one locus. And from here, we can measure the first order or main effect of a mutation from big A to little a by comparing the phenotypes of these two genotypes. This is the analogous figure for two mutations. So we have an A and a B, and we can measure the effect of, again, what I would call first order epistasis, what any biologist would call the main effect. I can measure the effect of here, the big A to little a mutation on the little b background that's shown at the top, or I can do that exercise on the big B background that's shown at the bottom. And the difference between those two estimates is where the pairwise epistasis lives. If you're troubled by the asymmetry in this figure, don't be. I could just as well measure the effect of the little b to big B mutation on the big A background, compare that to the little b to big B mutation on the little a background, and you can see very quickly, either by doing the math or counting degrees of freedom, that it's going to be the same pairwise epistasis term. So what I've done on the right here is after controlling for the main effects, I ask what's left. What's the discrepancy between main effects, and that's the pairwise effect. We do the same thing for a system with 3 mutations after accounting for the main effects, which are edges, and pairwise effects, which are faces. The third order effect is the discrepancy among faces across the cube. Similarly, in a 4-dimensional case, the fourth order epistatic effect is the discrepancy after accounting for edges, faces, and 3-dimensional cubes. And we can play this game ad infinitum. Now, the thing is, deep mutational scanning doesn't bring us into contact with phenotypes for most of the nodes in sequence space. As I said, it's an exhaustive sampling at radius 1 around 2 starting points, and we know nothing about what's going on in the middle. So how can we get from that kind of data to epistasis? And what I mean, again, is I want to think about, in a principled way, what kind of signal of epistasis, where should I look for signals of epistasis? So to illustrate my thinking, and my thinking is still very incomplete here, I'm going to imagine a toy story where TEM1 and SHV1 differ by only 3 mutations, and they are only 4 positions long. So on this 4-dimensional hypercube, I've located TEM1 on one node, and at Hamming distance 3, I've located SHV1. This, I think, is the smallest system that lets me start to get some traction on where the signal may lie. So the fuzzy red circles are the 3 genotypes at radius 1 from TEM1 whose phenotypes I'll know from the experiment. The blue circles are the same thing for the 3 single mutant neighbors of SHV1. And these 6 open circles are the beginnings of this huge space between these two spheres of radius 1 where I won't know anything the way this experiment is planned. So here is the representation of the method of analysis that leads to the scatter plot I showed you a few minutes ago. We're going to compare the effects of the same mutation on two backgrounds. So here I'm highlighting just the edges that correspond to a mutation at one of the positions where the two backgrounds are identical. And what we're doing is really this sort of double dashed line represents one cube that captures the space between the two starting sequences, which differ by Hamming distance 3. And there's a similar cube at the back of this hypercube that does the same thing for the two mutants. So that's like this, except instead of differing by 91 amino acids, which is the real experiment, the two backgrounds differ by 3 mutations only. Another way of going after this is to compare the effects of reversions between backgrounds. So everything I've said, I've been focusing only on those positions that are the same in the two backgrounds in TEM1 and SHV1. But in positions that differ, I can revert the amino acid that I find in SHV1 to the TEM1 state, and similarly revert the TEM1 position to the SHV state. And that looks like this. So here I'm converting between, I'm examining mutations that occur at positions that the two backgrounds differ at. And now I'm going to compare the representation of epistasis on this face with the representation of epistasis on this face. Okay, that's all I have to say. And as I said at the outset, this is something that I'm still in the early stages of wrestling with. I do feel that there's an opportunity to sort of take a principled perspective, starting from sequence space and thinking about where in sequence space the deep mutational scanning data will live to develop a strategy to extract the most amount of information possible. One point I'm concerned about is that there only are a finite number of observations. And I have the intuition that if one isn't careful, we're going to use the same observations and the same contrast between observations, thinking we're estimating more than one epistatic term when in fact they are intrinsically dependent on one another owing to the structure of sequence space. Okay, wrap it up. Paired deep mutational scanning libraries offer an opportunity to study epistatic interactions, and I'm really excited because this is an opportunity to study epistatic interactions before natural selection gets its hands on the alleles. And I'm desperate to understand the most effective ways of extracting information from these sparse samples. A point I didn't raise is all the sequence space pictures I drew assumed biallelic positions, but of course, amino acids have an alphabet of 20, which introduces some complexities that I really don't fully know how to work with. So with that, I'll stop. Thank you very much for your attention. I want to acknowledge Dave Morgan and Lauren Crawford again and the National Science Foundation. And I'd be very grateful for any questions.
Okay, thanks a lot, Dan, for the great talk. We do have some questions. I see also a raised hand. So Junichiro, would you like to ask a question?
Oh, sorry, I kind of missed, sorry.
Okay, so we have questions in the chat. So Valentin is asking, what would be the advantages or differences between your approach of mutating two different backgrounds and exploring the double mutations on one given background when exploring epistasis? Is there a reason why you chose one over the other?
Well, it's a matter of practicality, and I'm a little bit anxious that someone's actually developed the technology to build exhaustive sampling of radius 2 while I wasn't reading the literature. But the size of that experiment goes up very quickly, and to my knowledge, it's not possible to build that kind of data set. Moreover, we do come to the question that I asked about how far apart would we like to be, how far apart in Hamming space would we like to be making these comparisons in order to be able to learn the most?
Okay, great. We have another question from Michael Manhart. He says, I recall that there are also double mutant scans for some proteins. That only indicates pairwise and not higher order epistasis, but how would that data fit into your analysis?
Yes, thank you. And that question is really very closely related to the first question. The study that I'm aware of, which is perhaps the one you're thinking of, also came from Ostermeyer's lab. And what they did was to mutagenize adjacent pairs of residues. So they made all combinations of amino acids at position 1 and 2, and then at position 2 and 3, and then at position 3 and 4. And there's a very strong signal of epistatic interactions in those data, but it has this sort of primary sequence structure, structural bias, I would say. I think it's an open question of whether that is more or less informative than another approach, but that's the way I think about that.
Okay, great. Next, we have a question by Joachim Krum. Maybe you would like to ask your question? Or yes.
Yeah, I can ask it. So I think this idea of sort of systematically changing the distance between the two backgrounds is very nice. But the question is, to what extent is that feasible experimentally, or are you just stuck with, if you're like in the TEM1 case, you're stuck with two versions, and then the distance is what it is?
Yes, sure. I think both are true. If one could, I guess I would have to start by prefacing that I think big, but I'm actually, I do very small experiments. But it seems to me that if one did an exercise of this sort, let's say 3 times over, where the distance between the two backgrounds, the identity is 60%, 80%, and 90%, even though they're different systems, it might be quite informative in terms of learning something, not about the biology of those particular systems, but something about the way epistasis emerges over time as the focal sequences diverge from each other.
And just a comment, I think that conceptually this is related on the one hand to Luca Ferretti's gamma measure, right? Where you sort of look at the way a mutational effect changes as a function of distance. And also Christina Kroner has introduced something that she calls rectangular perturbation,
yes,
in paper, which I think is also sort of similar to what, looking at epistasis over larger distances between the backgrounds.
Yes, yeah, that's really good. Thank you, Joachim. Those are two people that whose work I haven't really thought carefully enough about in this context. So I appreciate that very much.
Okay, great. We have another question from Stephen. There is also data with mutations of all order within a randomized subpart of the sequence, Wu et al. 2017, Boyer et al. 2016, though clearly limited in length. Couldn't you use these anyway to study the first question on the optimal genetic background length?
I am endeavoring to find my way to the chat window because I didn't hear the citation.
Oh, sorry, it's Wu et al. Okay, yeah, it's Wu et al. 2017, Boyer et al. 2016.
Let's see. I'm maybe not familiar. There was a study from George Zhang's group where they mutagenized a tRNA in yeast is one of these. So what they did there was they had a solid state technology. So the tRNA is only 80 nucleotides long, and they had a solid state technology that let them mutagenize quite exhaustively out to, let's say, radius 3, and then a little bit more sparsely beyond that. And we've actually looked at those data, which are of course quite interesting. The limitation at that time had to do with this solid state technology which is only applicable to genetic segments that are sort of order of 80 or so nucleotides long. But I don't know whether I'm answering the question or not.
Yeah, I mean, there was a part about the limitation in length indeed. The rest of the question was couldn't you use these anyway to study the first question on the optimal genetic background length?
Oh. I hadn't thought about that. So I am jotting these down. And Stephen, if you could send me full citations by email or in the chat window right now, I'd be very grateful, because I'm not sure I know either or both of these papers.
Okay, yeah, I mean, okay, good. I mean, let's take this question for later. And so next, Martin had a question. Martin, I saw your webcam on before. Would you like to ask it directly?
Yeah, I can ask. Hi, thanks for the talk. So my question was just, you mentioned at the reverting mutation. I was already typing a long question, which I canceled because I wanted to ask about this. So did you actually look in Bloom's data to the reverting mutations?
We haven't done that yet. Yeah, no.
Because obviously there's much less statistics because there's only one variant and not 19, but you had 60 of them. So might be interesting. As you did.
Absolutely. Yes, just haven't done it.
Okay, this one.
The truth is, understanding how to talk about and implement the scatter plot came much more quickly to us. So we've pushed a little bit harder on that line of analysis. Then I had this other idea of looking at the positions that that are different. And then I got very anxious about the thing I mentioned at the end, which is I'm worried that if I'm not careful, I'm going to believe I'm learning more than I can because there really is such sparse sampling. And so that's when I stepped back and thought, let me try to take a principled approach to understand how much data, how much signal really lives in these kinds of data sets. But yours is a very reasonable question. Just to look, it would be fun.
Okay, great. We have a last question from Daniel Fisher. Daniel, would you like to ask your question live or should I read it?
Yeah, I guess I'll ask that. Sorry, I missed the beginning of the talk, so maybe you said this, but I guess I'm a bit confused about why one wants to expand in the orders of epistasis once you've got large numbers of mutations involved. And presumably one is trying to get statistical information of how the difference in effects of particular mutations differ as a function of the genetic distance and things between them. And I'm not sure whether the representations that at least I saw you using are natural for that.
Me either. No. It's a very fair question. And we spent some time. I and my collaborators spent some time thinking quite explicitly about interactions with different orders. I don't think the kind of experiment that we're doing is going to be very informative about interactions of particular orders. It's just a framework for me to remain focused on things that are more complicated than pairwise. That's not a very...
Yeah, one can more generally ask about the statistics of effects as a function of the genetic distance and how you lose correlation in the effects of a particular mutation as the sequences, the backgrounds get further and further apart, and how you lose that and so on. And I think it's is maybe a framework that's sort of easier to get at some of the statistical questions.
Yeah, that seems reasonable, Daniel. Thank you.
Okay, great. I mean, so I think because we are already a bit late in the schedule, we should stop the discussion here, or rather move it to the Gather space. So yeah, we have our social in Gather, and so we should